Hello, welcome to another screencast where this time we'll be continuing a discussion of nervous system and membrane potentials. And this time, first I need to find my pen, here we go. This time we'll be talking here, I think we need to spend a minute and do this, right? Talk about facilitated diffusion and active transport. And then we get into this activity, the sodium-potassium pump. And then for a third screencast there, we will look at the membrane potential. And again, we've already looked at in a previous screencast A and B here. So again, we're going to focus right here on this, what it means for facilitated diffusion and active transport. So let's get started here. As a review, you need to understand that the membranes of cells are made of phospholipids, and these phospholipids have hydrophilic heads and then hydrophobic tails. And going back and looking at your notes for the structure of a phospholipid is critically important to understanding this. Right? So you do want to do that. Knowing that the membrane here is not just a phospholipid bilayer, but that there are proteins associated with that as well. And these proteins are often involved with transporting molecules across the membrane. So in diffusion versus facilitated diffusion, a reminder here that diffusion, both of these are diffusion, both of these processes are diffusion. This is how things get across a cell passively. So this is passive diffusion, and the definition of diffusion is a random movement of molecules. Right? So these molecules in solution. If you don't know what a solution is, you should go back and review that. Right? So in solution, you have these molecules that are dissolved. And so if you don't know what dissolved is, you should go and figure that out as well. So these molecules that are dissolved are moving randomly, and here we see them in a high concentration outside of the cell. Right? So their concentration in brackets here is high outside the cell. The concentration of those same molecules in the solution inside the cytoplasm is low. And so because these molecules we're showing them moving right across the membrane here, these molecules are small, these molecules are non-polar, right? And they are also non-charged, uncharged is a better word there. So uncharged. And really the only examples we have of things that will go across the membrane most likely are going to be oxygen and carbon dioxide gas. Right? So these things will diffuse straight across membranes. Turns out, though, that most substances don't fit, most solutes don't fit that description. So here we have a solute down here that might be large, polar, right, and or charged in some way. Right? And so examples that we have of these things would be ions, would be something like glucose, and proteins as well. So these things, even though their concentration is high on this side of the membrane and is low on this side of the membrane, this favors the movement of them inside the cell, in this case, and moving down their concentration gradients. They will not do so because of this phospholipid bilayer right here. And so what we have is some sort of protein channel or some sort of protein transporter that will facilitate, right, or help get those molecules across. But it's still diffusion because they're moving from an area of high concentration to an area of low concentration, an area of high concentration to an area of low concentration, until there's a dynamic equilibrium. I want to caution you, right, that when you write about this, when you talk about this, you should not be saying that these purple dots need to move in, or that they want to move in, that these yellow triangles, orange triangles need to move in or want to move in. There's no needing or wanting here. They just will because of this random movement of these molecules in solution. Right? So let's try to not anthropomorphize these things too much here. Right? So again, these are all passive transport mechanisms, again, going from an area of high concentration to an area of low concentration until there is a dynamic equilibrium established. Right? That dynamic equilibrium, of course, would be for every one molecule that moved across, the other one would move back. Right? So you should pause there and get that straight. Here's another example of diffusion, not across a membrane, but over here, not across a membrane. but then here we do have it across the membrane. Maybe run the slideshow and click on these if you're having a hard time with diffusion versus facilitated diffusion. There's another example of facilitated diffusion. Pause. Take a look at those diagrams, see why they make sense. Look at the difference between a channel protein here. What is this similar between a channel protein and then a carrier protein or a transporter protein? Good opportunity to review here. Let's talk about active transport. Active transport has a specific definition here, and this active transport is going to be a method of transport of getting molecules from an area of low concentration to an area of high concentration. So what we say here is that this active transport, again, these things are probably large. These solutes are probably large, charged, or polar molecules. And if you don't understand polarity, this is also a good time to sort of go back and review what we mean by polar. So I was saying that you're moving these from a low concentration to a high concentration. And this, again, this is going to be against a gradient. And let's say that so this is presuming that we already have a gradient established, right? But you can also use active transport mechanisms to establish a gradient. So to make a gradient, to make this concentration difference, right? And so again, reminding you that a gradient is a concentration difference, and that concentration difference is described as being across a membrane. So we've got this concentration difference across the membrane. On one side of the membrane, we have a low concentration. On the other side of the membrane, we have a high concentration. Right? So we're going across the membrane here. So we're going to be comparing here and here, comparing side A with side B. So there's a gradient established if there's a difference in the concentrations of these things across the membrane. Now, what we're seeing in these active transport mechanisms is that ATP is often used as an energy source to move these molecules against the concentration gradient. But it's not only ATP all the time. We've seen earlier in the year that moving electrons through an electron transport chain and the series of redox reactions will also provide a negative delta G to run these transport mechanisms here. Okay, so you should stop here, pause, and refresh your memory. Practice, get your confusions down about active transport versus facilitated diffusion, because those concepts play key roles in understanding membrane potential and how the neuron works. Okay, thanks for listening. Bring your questions to class.